Hello, good morning. Good morning, everyone. Am I audible? Sound is coming? What's happening? Check. Okay. So, in last class, we discussed up to endomembranous system, isn't it? Right. So, today we are starting with endoplasmic reticulum. So, if we talk about ER, as you know, it is a network of reticular structure present in cytoplasm. When you observe a cell with the help of electron microscope, you will find in the cytoplasm there is a network is present, and this network is made up of a reticular structure, and this reticular structure is called endoplasmic reticulum. If you look the diagram of it, we will got the better clarity. Let me draw somehow bigger diagram. Suppose that this is a cell. So this one is plasma membrane. And here it is nucleus, double membrane bounded structure, and you know nuclear membrane. And if you talk about the nuclear membrane, so this nuclear membrane is discontinuous at places, and that is the reason we are telling who is present. Nuclear pores are there. So this is the nucleus. Now what happened? Just try to understand. From the outer membrane of the nucleus, a network of tubular structure assemble like this. And this is called endoplasmic reticulum. Everywhere, all around you will find this endoplasmic reticulum is present. This side, everywhere you find these nuclear structures are there. So one side ER attached with plasma membrane. Are you getting the idea or not? One side endoplasmic reticulum attached with plasma membrane. Another side endoplasmic reticulum attached with nuclear membrane. Right. So it is a network like a structure present. Clear? Is it all right? If you find about the new endoplasmic reticulum, somehow observe it more closely with more magnification, you will find the endoplasmic reticulum who are reticular structure who are near the nuclear membrane, are which one? Rough. And there is a granular depositions. And these part is called RER, rough endoplasmic reticulum. And toward the plasma membrane, this granular deposition is not there. And this part is called SER, smooth endoplasmic reticulum. Is it clear? It is all right. So if we talk about ER component of ER. So three types of components are present in the ER. First, I am writing cisterni. Second, I am writing vesicle. And third, I am writing tubule. So these are the three different components of the endoplasmic reticulum. If you talk about this cisterni, it is a flat structure and arranged in a stack. So look, these are the structure, flattened and arranged in a stack. These are called cisterni. So what about cisterni? This is cisterni. It is flattened, flat structure and arranged in a stack. Arranged in a stack. One above the other. It arranged like this. If you talk about vesicle, vesicle is somehow a round or oval structure. These are the vesicle. It is oval or round, a spherical structure. And tubule, if you talk about tubule, it is a branched tubular structure. Branched tubular structure. So these are the three components. These are the three components present in which one? Endoplasmic reticulum. These tubules look like this. You just think, have a look. It is a tubule. You are finding it is a branch tubular structure. So this is cisterni, this is vesicle, and this is the tubule. So there are three components of the endoplasmic reticulum: cisterni, vesicle, and tubule. Are you getting the idea? Diagram I have drawn: cisterni, vesicle, and tubule. Is it clear in the mind? All right. Now you just observe the endoplasmic reticulum diagram here. You just observe the endoplasmic reticulum diagram here. Whenever you observe the diagram of the endoplasmic reticulum, what you are finding? This endoplasmic reticulum is dividing the cytoplasm into two parts. Getting the idea? This side, every side endoplasmic reticulum is there. So they are dividing the cytoplasm into two parts. One, look, I am just putting a point here. So this part means it is called intraluminal. Intraluminal means these are the cytoplasm who are present inside the lumen, inside the pipe, tube of the endoplasmic reticulum. You can say intraluminal cytoplasm. Are you getting the idea? I am just magnifying this diagram somehow more. You look here, this part is present inside the lumen, inside the lumen of ER. And this part you can see. This part is not in the lumen. This part is not in the lumen. These are in lumen, these are in lumen, but this part is outside of the lumen. Is it or not? And this part is called extraluminal. So you got the idea? Intraluminal and extraluminal, is it or not? Tell me, hurry up. Right? Intraluminal is what? The cytoplasm present inside the lumen of the endoplasmic reticulum, and extraluminal who are present outside the lumen. So it is dividing the entire cytoplasm into two components, intraluminal and extraluminal. See na, how much membranous network are there? It is membranous network everywhere in the cytoplasm. You can say like about 50% of membranous structure present in the cell is alone occupied by whom? Alone occupied by whom? Endoplasmic reticulum. Is it clear in the mind? See, everywhere extensive membranous structures are there. Is it clear? So now we are going to talk about the type of endoplasmic reticulum. Okay, ji? So as I told, there are two types of endoplasmic reticulum. Have a look here. If we talk about ER, endoplasmic reticulum, this endoplasmic reticulum is of two types. One is RER, rough endoplasmic reticulum, and another is SER, smooth endoplasmic reticulum. So RER, rough endoplasmic reticulum, who is present? Ribosome present. Ribosome present in rough endoplasmic reticulum, and in a smooth endoplasmic reticulum, ribosome absent on the surface. Ribosome absent. And where ribosome present? It is present on the surface of ER. How ribosome is present? Let me tell you. Suppose that this is the surface of the ER. Let me draw somehow down. This is a surface of ER. These are the surface, upper surface. So you will find ribosome present like this. This is 60S ribosome and this is 40S, a smaller subunit, 40S ribosome. 60S, 40S, 60S, 40S. Eukaryotic cell, na? So 80S type of ribosome is present. How many, which type of ribosome is present? 80S type of ribosome is present. Here it 80S ribosome present on surface of ER. And these ribosome, when attached, someone act as a glue, na? So it is called ribofurine. Ribofurine. Ribofurine, two type of ribofurine, 1 and 2 are present. And what this ribofurine is doing, this ribofurine helping the larger subunit of ribosome to join with, suppose that this is the surface of ER and this is the ribosome. So for joining this, something required, na? So ribofurine is required for holding these two. So ribofurine acting as a glue here who help in attachment of ribosome as well as surface of the ER. Getting the things or not? So due to presence of these ribosome, these surface is appearing, which one? Rough. And that is the reason we call this endoplasmic reticulum as what? RER. Here who is absent? Ribosome surface, no granular structure are there. No granular structure are there. Ribosome is absent. And here also ribofurine 1 and 2 is absent. Getting the idea? So what I am telling, you got these two differences still now between RER and SER or not? Yes, it is clear. Here ribosome and ribofurine absent in SER and ribofurine and ribosome are present in RER. And how the ribosome attach with the ER? With larger subunit or a smaller subunit? It attach with which one? Larger subunit, 60S. Are you getting the idea? Is it clear? Now I am telling some more differences between rough endoplasmic reticulum and smooth endoplasmic reticulum. So if you talk about RER, it consists mainly of cisterni. I told na, in endoplasmic reticulum, three components are there: cisterni, vesicle, and tubule. So in rough endoplasmic reticulum, more part is made up of which one? It is mainly made up of which part? Cisternal part. You can getting the idea. It is consist mainly of cisterni, or you can say cisterni is present in more amount. And here major component is what? Here major component in ACR, it consists mainly of tubular part, mainly of tubules. Mainly of tubules. Getting the thing? This doesn't mean another part is not there. Another part also present, but cisternal part is more in RER and tubular part is more in smooth endoplasmic reticulum. If you talk about RER, it is toward nuclear membrane. Because everywhere reticular structure is there, no? So it is toward nuclear membrane. Toward nuclear membrane. If you talk about ACR, it is toward plasma membrane. It is toward plasma membrane. I already told it one side attached with the nuclear membrane, another side attached with which one? Plasma membrane. So toward plasma membrane, who is there? Toward plasma membrane, a smooth endoplasmic reticulum is there, and toward nuclear membrane, rough endoplasmic reticulum is there. So it is present abundantly in cell participate in protein synthesis. Participate in protein synthesis. Who abundantly present in protein synthesizing cell? Rough endoplasmic reticulum. Because you better know na, ribosome is present on its surface. And ribosome is called protein factory. So protein synthesis occur on ribosome. Protein synthesis not occur on ER, don't confuse. If someone asks you a question, protein synthesis occur on ribosome, ER, what the answer will you give? Ribosome, not occur on ER. Why we are saying ER? Because ribosome present on the surface of ER. So who are giving the sitting space for ribosome? ER. Able to understand? So wherever RER is there, who will present? Ribosome is present. Is it clear or not? So that is the reason I am writing here. It is present abundantly in cell participate in protein synthesis. ER itself not synthesize a protein. Many times people confuse. ER function, they write protein synthesis. RER function, protein synthesis. It is wrong to say. RER function is not protein synthesis. RER associated with protein synthesis. Though both are different words. Function protein synthesis means what? RER itself synthesizing the protein. Actually, protein, it is rough surface. And rough surface have which one? Ribosome. And that ribosome present on the rough surface are synthesizing the protein. Are you getting the idea or not? Now, if we talk about the ACR, smooth endoplasmic reticulum, it is present abundantly in cell associated with lipid synthesis. Lipid and steroidal hormone synthesis. Steroidal hormone synthesis. Getting the idea? So where RER is present, where ACR is present, you got the idea? RER present in cell who are involving in protein synthesis, liver cell, so many enzyme form. Getting the idea or not? Right? If you talk about lipid synthesizing cell, able to understand? A steroidal hormone synthesizing cell, who is present abundantly? Who is present abundantly? ACR is present abundantly. All the sex hormone, testosterone, estrogen, these all are which kind of hormone? A steroidal hormone. Getting the idea or not? So the cell who are associated with synthesis of these hormone, where who is abundantly present? Who is abundantly present? Smooth endoplasmic reticulum abundantly present. Clear? It is all right. You got the difference between RER and SER. It's over, done. Now I am telling about function of endoplasmic reticulum. A structure we have seen, component we have seen, type we have seen already. Now we are going to come on which one? Function of ER. Function of endoplasmic reticulum. Have a look now. So if we talk about function of endoplasmic reticulum, you just write RER associated with protein synthesis. Protein synthesis. Okay. Second function I am writing SER. It is associated with lipid and steroidal hormone synthesis. Third function that is written in NCERT, so I write first in the priority order. Third function of ER I am writing, it increase the surface area of cytoplasm. And hence have a role in metabolism. It increase the surface area of cytoplasm or not? Many tubule-like structure, so many enfolded structure, so surface area increase, so site for metabolic activity increase, so playing a role in metabolism or not? More surface, more metabolism. Fourth function, it acts as cytoskeleton. Function of cytoskeleton is what? Microtubule, microfilament, intermediate filament, what? A structural shape, support, such kind of function are there. It is also provided by ER. You look now, this is cell membrane, this is nuclear membrane, and these are what basically? These are endoplasmic reticulum. One side attached with nuclear membrane, another side attached with which one? Cell membrane. So have that structural support or not? So it is working like it is a cell organelle who are working like a what? Cytoskeleton or not? Getting the idea? Is it clear in the mind? Now I am writing another function. Another function. I already told you कि it is a network like a structure. One side attached with nuclear membrane, another side the cell membrane. So it is responsible for intracellular transport. Intracellular transport from one part of the cell. Suppose that anything want to move from nucleus to the cytoplasm, they can pass through the pipes of the endoplasmic reticulum or not. So intracellular transport is another function associated with ER. Another function, ER also have a role in sending something out of the cell. Why? Because it is also attached with which one? Plasma membrane. Understanding the idea or not? So intracellular transport it is doing. Is it? Now I am writing some more function of here. Folding of protein. Folding of protein. When protein synthesis occur, it occur in which one? Primary structure of protein is forming, na? N-terminal, C-terminal. N-terminal, C-terminal. Now, when protein undergoes in tertiary structure, it going to enfold it like this. So these folding of the protein occur in which one? Endoplasmic reticulum. Another function I am writing, glycosylation. I am putting a star mark here. Glycosylation. It is written in NCERT in Golgi body. The formation of glycoprotein and glycolipid. Getting the things or not? Formation of glycoprotein and glycolipid occur in Golgi body. It is written on chemical modification, Golgi body associated. ER also associated. But in NCERT, who is written? Golgi body only written. I am telling you, if you have question bank, go for 5-6 year back question. You will find a question came, glycosylation occurring. Glycosylation occurring. And who is not in the option? Golgi body not in the option. Golgi body not in option. Getting the idea? Who was in option? ER is in option. If you have question bank, you just go through that question. So if you not have knowledge of this line, you're just going to mistake it or not. Is it clear? Understand? And that is the reason I wrote glycosylation. Also, you not confuse, so I'm writing also performed by endoplasmic reticulum. Now you just be wise. If a question is coming, ki glycosylation done by, option is there Golgi body, ER, both. We choose the both. If option is there Golgi body, ER, both is not there, so we choose Golgi body because it is written in NCERT in Golgi body. Getting the idea or not? So we have to be wise, we have to handle the question. And if not Golgi body is there, of course we have to choose which one? ER. It is also done. Getting the things? Another functions I am writing. Another function. So another function of ER I am writing is formation of Golgi body. Formation of Golgi body. Golgi body and micro bodies. Not only Golgi bodies, micro body also formed by whom? ER. Like peroxisome, glyoxysome, spherosome. These all are formed by which one? ER. Ninth function, it have a role in formation of lysosome. Formation of lysosome. Getting the things? Have a role in formation of nuclear membrane. Nuclear membrane. Endoplasmic reticulum also have role in formation of nuclear membrane. Now I am writing another function. Formation of phospholipid. Phospholipid, ER is associated. Phospholipid formation occur in which one? Endoplasmic reticulum. Now I am writing another function. Detoxification of drug. Detoxification of drug. Pollutant. Allergent. Occur by a smooth endoplasmic reticulum. Who is doing the detoxification of drug? A smooth endoplasmic reticulum. Getting the things? Actually, there is an enzyme present. That is cytochrome 450. This is responsible for detoxification. This is responsible for detoxification of a drug. So are you getting the idea? So these are the functions of the endoplasmic reticulum. So endoplasmic reticulum have a role in formation of Golgi body, formation of lysosome, formation of nuclear membrane, formation of which one? Microbodies. Is it clear in the mind? It have role in detoxification of drug. Getting the things, intracellular transport, giving the network like a structure, and most important, what I wrote, what is written in NCERT, and who is going to come in most of the test. That is what? That is what? In the very beginning I wrote, na? That is associated with RER associated with protein synthesis, RACR associated with steroidal hormone synthesis and lipid synthesis. That is the most time will come in the papers. Because we believe one thing, you teach many things, but give what is written in NCERT for practice. And why we are teaching it? To handle that kind of question. If I not taught, how can you handle? Golgi body not given. NCERT only wrote glycosylation occur in Golgi body. In paper, Golgi body not in option. Who is in option? ER in option. And usually these two cell organelle, endoplasmic reticulum and Golgi body, where examiner cross the boundary of the NCERT. Getting the things or not? Right? Is it clear? Done? Now we are going to move on next cell organelle, Golgi body. Golgi body. Right. Already you know who is forming the Golgi body. Endoplasmic reticulum. Who is forming the Golgi body? Endoplasmic reticulum. Right? Done? Now I am moving ahead about the Golgi body. So, discovered by C. Golgi as dense material near nucleus, and in honor of his name, getting the things or not, we just give its name as what? Golgi body. Is it clear? So Camilo Golgi. So as a dense material, dense reticular material, you can say dense reticular material near which one? Nucleus, right? If you talk about Golgi body, it also have three components. Because it evolved from who? Evolved from ER. So like ER, it also have three components. Cisterni, second component vesicle, and the third component tubules. Is it clear in the mind or not? So they have three components like what? Cisterni, here also you will find somehow flat, it is disc-like a structure. It is cisterni. It is somehow concentrically arranged, na? And they are interconnected also. Getting the things? So these are cisterni. So what is the diameter of the tube? NCERT wrote diameter of the cisternal tube. What is the diameter? Carry on. Very good. What 0.521 nanometer, micrometer, what? Micrometer. Yeah, good. So 0.52, and there is a dense material. There is a dense material present in the cisterni, and these dense material which is present in the cisterni is called nodes. They are called nodes. Getting the things? So if you talk about these are vesicle, and I am also drawing what? Tubular structure. This is tubule. It is present like this. I am making some more vesicle. Some more vesicle. So these are the Golgi complex. How ER and Golgi body differ? How they are differ? Three component is present in ER also, Golgi body also, but there is a difference. In ER, these all three components are scattered. And in Golgi body, all three components are assembled together. They are assembled together. Are you getting the idea or not? Understanding the idea or not? So how the arrangement of these three structures differ in all three, you got the idea? In ER, they are not attached, separated, and here they are already assembled. And that is the reason Golgi body is also called what? Golgi complex. Complex. It is made up of three type of structure: cisterni, vesicle, tubule, and these all assembled together. Is it clear in the mind? Now I am drawing the Golgi body structure along with the cell. Then you got a better picture, right? So have a look here. So look here, this is a cell. This is the nucleus. And now here I am drawing Golgi body. This is Golgi body. Are you getting the idea or not? Here it is the nucleus. This is plasma membrane. And this is Golgi body. Are you getting the idea? Now what I am finding, in Golgi body you look, it is like this concentrically type shapes. So this face of the Golgi body, who are toward the nucleus, two faces there, na? The face of the Golgi body who is toward the nucleus is called cis face. It have another name, forming face. Forming face. This cis face is convex or concave? It is convex. Convex. So the convex side of the Golgi body is present toward the nucleus. This is called cis face and forming face or convex face. Getting the idea? And the concave face of the Golgi body, who are toward plasma membrane, concave, it is also called maturing transphase or maturing phase. Maturing phase. So this Golgi body is a cell organelle with polarity. Is a cell organelle with polarity. I am telling one as a cis phase, another as a trans phase. One as forming phase, another as maturing phase. One is convex phase, another is concave phase. Are you getting the idea? And that is the reason we called it is a cell organelle with polarity. It is a cell organelle with polarity. And both the phases are different but interconnected. Both phases are different but interconnected. So you just look here, this is interconnected with this, here interconnected with this. Getting the things or not? So cis and trans phase are interconnected with each other. Both are different to each other, but they are interconnected to each other. Are you getting the idea? Understanding the idea? Now what happened? Just try to understand. Have a look here. Around the Golgi body, in the cell, usually very less cell organelle or no cell organelle are present. Getting the things or not? Okay, ji? And that is the reason we called it Golgi body, that is zone of exclusion. So if someone going to ask you a question, what is zone of exclusion? Zone of exclusion. What is the zone of exclusion? So you can write like around Golgi body, there is no, there is less or no, there is less number of or no cell organelle present, just nearby the Golgi body. This area is called zone of exclusion. This is called zone of exclusion. This is called zone of exclusion. Are you getting the idea? What is zone of exclusion? Clear in the mind? Around the Golgi body, there is less number of cell organelle and no cell organelle is present just near periphery of the Golgi body, and this area is called what basically? Zone of exclusion. Is it clear in the mind? What is zone of exclusion came in the mind or not? Now I am telling you about the function of the Golgi body. So cis phase you understood, trans phase you understood, clear? Convex is cis or forming, getting the things or not? Right? Is it clear? Right. Now give your attention. I am writing the function of Golgi body. Function. Function of Golgi body. Secretion. Cell secretion. Second function of a Golgi body. Packaging. Third is chemical modification. I am writing first in priority who is from NCERT. Chemical modification. Example, formation of glycoprotein and glycolipid. Formation of glycoprotein, protein joined with carbohydrate. Who is forming? Glycoprotein and glycolipid. When carbohydrate joined with lipid, it is called glycolipid. This chemical modification is occurring which one? Golgi body. And these things is called what basically? Glycosylation. So about glycosylation, who is the cell organelle written in NCERT? I already told it is written is what? Golgi body, but it also occur in endoplasmic reticulum. Able to understand? Now, Golgi body, it is also called middleman of cell. Middleman of cell. Or it have another name also, or director of macromolecule traffic. This is another name of the Golgi body. Golgi body associated with formation of lysosome. Formation of lysosome. Golgi body have a role in cell wall formation. Cell wall formation. Golgi body have a role in lysosome formation. I told Golgi body have a role in which one? Cell wall formation. Getting the things or not? Have a role in Acrosome formation. Acrosome formation. Getting the things? Is it clear? So, these are the some of the function associated with which one? Golgi complex. So what the function I wrote, let me somehow minimize it and I will explain it also, don't worry. Function I wrote, secretion, packaging. What is secretion? What is packaging? I will tell you. Chemical modification. That is glycosylation, formation of glycoprotein, glycolipid. It is called middleman of the cell. Why called? I will tell. It is also called director of macromolecular traffic. Why called? I will tell. Able to understand? You can say about it also called postmaster of the cell. It is also called what? Postmaster of the cell. Why called? I will tell it. Formation of lysosome have a role in cell wall formation. Getting the things or not? And because it have associated with lysosome formation, so also have a role in which one? Acrosome formation. So these are the functions of the Golgi body. Now actually look what happened, what actual story occur. Try to understand what actual story occur. Should I change this slide? Should I change this slide? Are you getting the idea? Yes. Actual story occur is, this is the cell. This is the nucleus. This is what? ER. With nuclear, it is what? Endoplasmic reticulum. This is endoplasmic reticulum. Okay, now I am drawing, this is what? Golgi complex. Why we called endomembranous system altogether, you will understand here. Okay, ji? Got the idea? Now, look here. Here on the ribosome present on RER, ribosome present toward the nucleus on ER is forming protein. What it is forming? Protein synthesis occur on ribosome. Now this protein entered in the lumen of whom? These protein enter in the lumen of whom? Endoplasmic reticulum. Clear? Connectivity is there or not? These protein enter in the lumen of which one? Endoplasmic reticulum. After that, entering in the lumen of endoplasmic reticulum, these protein undergoes in a structural packaging. Protein undergo in folding and primary structure converted into secondary structure or tertiary structure. So folding of protein occur inside the ER. You got the idea? Understand the idea? Clear? Done? Over? Is it? Now, these membrane is going to pinch off. I'm using a word, who is going to pinch off? These are going to exvaginate and membrane is going to pinch off, and these are going to keep which one? Enfolded protein. So this is called vesicle of ER. Vesicle of ER. ER also have vesicle or not? So this is called vesicle of ER. What happened? Membranes is going to exvaginate, there who enter? Protein enter and then it cut. So it becomes separate. Now this vesicle of ER gets separate. Now vesicle of ER migrating this side. This is vesicle of ER. This is vesicle of ER. Let me take the another color. This is vesicle of ER. And in this vesicle of ER, who is there? Enfolded protein are there. Enfolded protein are there. Getting the things or not? Now it is which phase? It is the cis phase. It is the cis phase of the Golgi body. Getting the idea or not? It is the cis phase of the Golgi body. Now these vesicles fuse with the cis phase. These vesicles fuse with the cis phase. And when they fuse with the cis phase, they release their protein into the which one? Golgi body. Now these Golgi body, cis phase to transphase, this protein migrate, because both cis phase and transphase are connected na, so it is going toward transphase. It is going toward transphase from cis phase, and meanwhile when it is transporting from cis to transphase, who is going to occur? Here chemical modification occur. And suppose that in chemical modification, who attach here? This is a oligosaccharide. These oligosaccharide attach here. And after attachment of the oligosaccharide, it is converted into which one? Glycoprotein. These converted into glycoprotein. So who occur? Glycosylation occur. Who occur? Glycosylation occur when it is moving from cis phase to transphase. Connectivity is there or not? So look, protein form on ribosome, then protein enter in ER, then enfolding of protein occur in ER. After enfolding, these ER pinch off, these protein packed in a vesicle. After that, these Golgi ER vesicle fuse with cis phase. Protein transfer in Golgi body. Then it is migrating toward transphase. And when it is migrating toward transphase, chemical modification occur and glycoprotein form. Now later on from transphase, who will pinch off? From transphase, who will pinch off? Vesicle get pinch off. Here it will pinch off, and these protein, let me change the color, it pinch off, and these protein is present in these vesicles. This protein is packed in this vesicle is the better term. So here this modified protein is packed here. So here vesicle. In vesicle who is packed? Glycoprotein is packed. Chemically modified protein is packed. Are you getting the idea? Is it clear? So this is which vesicle? Golgian vesicle. Golgian vesicle, and it is vesicle of ER, because vesicle present in both ER and what? But ER vesicle carrying unmodified chemical, Golgian vesicle carrying modified chemical. Are you getting the idea? So chemical modification occur or not? So now it is a protein, it is an enzyme. Now where this enzyme will go? Where this enzyme will go? These enzymes go either at intracellular target or outside the cell. Suppose that likewise many enzyme form, many vesicle form. Try to understand the mechanism. Another vesicle, and every vesicle have protein or chemically modified protein or chemically modified lipid. Are you getting the idea? In such a way many vesicle will form together or not? Yes. Now you try to understand. You just give your attention. You got the entire picture. Have a look here. Suppose that here is mitochondria. So some of the vesicle go and fuse with mitochondria. So these vesicle, these Golgi body responsible for transport of which one? Proteins or chemically modified protein at intracellular target also or not? How they are getting enzyme? Some of the enzyme also go from the cytoplasm here, na? Control is there, na? All the enzyme not formed by mitochondria itself, na? So these are some plastid enzyme going there. Able to understand? When these are going to fuse with plasma membrane, these chemical from plasma membrane secrete. It is called cell secretion. It is called cell secretion. So who is going where? Which you think like these all vesicle is equivalent to later. Which later will go there? Which target? Which house? Either outside of house, outside of city, inside of the city, getting the things or not? So these are all duty performed by which one? Golgi body. So which macromolecule went where? Who will go where? So it is acting as a middleman. Na? Supply line check. You go there, you go there, you go there, you go there. So it is acting as a what? Middleman of the cell. It is acting as a director of macromolecular traffic. It acting as a which one? Postmaster. Are you getting the idea? Which letter will go there? Is it clear in the mind? Understanding the idea? And their function is coordinated or not? Their function are coordinated or not? Yes. Getting the things? Now I am telling in one line how lysosome is forming. How who is forming? Lysosome is forming. Many vesicle fuse together. Vesicle, 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 vesicle. Many Golginian vesicle fuse together and forming a structure, have many different type of enzyme. What basically? Lysosome. So lysosome, how it formed from Golgi complex, you got the idea? Understand the idea? Clear in the mind? Many Golginian vesicle fuse. Everyone have different enzyme. So lysosome have many different type of enzyme or not? Is it clear in the mind? Is it clear? It is all right. So lysosome formation mechanism you also got. Is it over? So ER, Golgi body, lysosome are very coordinated, na? And that is the reason we use a term. We use a term GERL. What is the full form of? Golgi body which arise from ER form lysosome. Golgi body which, why I am writing with, which arise from ER form lysosome. So Golgi body over. Now we are going to deal about which one? Lysosome and vacuole. Okay, ji? Lysosome. We are starting about lysosome. Give your attention. Lysosome formed from Golgi body. Getting the things? Due to fusion of many Golginian vesicle. Suppose that this is a lysosome. You know, better know, it is a single membrane bounded structure. So these lysosome contain hydrolytic enzyme. What is there? Acid hydrolases. Example, carbohydrates. It digests carbohydrate. Lipase. It digests lipid. Protease. It digests protein. Getting the things? So you can talk about more than 50 types of hydrolytic enzyme present in which one? Lysosome. More than 50 type of hydrolytic enzyme. Some of the example I told here. Able to understand. So if you talk about lysosome, work best at acidic pH. Because here enzyme is what? Acid hydrolases. So work best at acidic pH. Getting the idea? Understanding the idea? And around how much pH? 5 pH. pH around 5. It work best. Understanding the idea? Getting the idea? So if we talk about lysosome, it work at best at which pH? Acidic pH. Now question arises, how it maintain the acidic environment? So this is the suppose that cell, and this is assume what? Lysosome. Here it is what? Cytoplasm. So in the membrane of the lysosome, there is a pump protein present. And these pump protein actively pumping of proton from cytoplasm to which one? Lysosome. And in that who expenditure occur? ATP converted into ADP. So how a lysosome continuously maintaining a acidic pH? Are you getting the idea or not? Lysosome. See, I wrote lysosome. Got the idea? Understanding the idea? Is it clear? All right. So how the pH become acidic? By active pumping of the proton. Because when proton will go, concentration of H ion will increase. When concentration of proton ion will increase, pH will decrease. You have studied na? In zoology also, chemistry also. So how they are maintaining the acidic pH? You got the idea? Understand the idea? And it is a, I am telling about lysosome, polymorphic cell organelle. They have different structure at different physiological state. They have different structure at different physiological state. Physiological state. They have different structure at different physiological state. So what are the different structure at different physiological state? I am telling you. First is primary lysosome. Second is secondary lysosome. It is of two types. Autophagosome and heterophagosome. Third is tertiary lysosome. It is also called residual body. And these lysosome, if you talk, primary lysosome, here enzymes present in inactive form. Inactive form. Got the idea? Enzymes present in inactive form. Now I am telling about how primary forming, how secondary forming, how tertiary forming. Getting the things or not? I am telling you through a diagram. Just give your attention. You just have a look here. Suppose that this is a cell. And this is the primary lysosome. You know now how it formed? It formed by fusion of many vesicles of Golgi body. Agreed? And it contained more than 50 hydrolytic enzyme. So this is primary lysosome. I am writing, it is which one? Primary lysosome. Now what happened? Now what happened? All the enzyme in primary lysosome present in inactive form. This is a cell. Suppose that this is a amoeba cell. This is a amoeba cell, assume. Now here food is there in the amoeba. So this food is captured by amoeba, na? You just try to understand. Food is captured by amoeba. And when food is captured by amoeba, this food will come in and present inside a vacuole called food vacuole. So this is the food. This white color block I am making here is food. And this is food vacuole. So food vacuole. And this is which one? This is primary lysosome. Now question arises, inside the food vacuole, food is there. In amoeba, intracellular digestion occur. Who do this intracellular digestion? This primary lysosome do the intracellular digestion. How? Look here. These two will fuse together. These food vacuole fuse with lysosome. You just have a look. This food vacuole fuse with the lysosome. Here is the food. And who will release on the food? Enzyme will release on the fuse or not? So its structure or its structure are same or different? Different, na? So this structure is called secondary lysosome. And it is also called which kind of secondary lysosome? Heterophagosome. It is eating who is not a part of body, who are eating, it is eating different things, not in own component, own cell component. So it is heterophagosome. So heterophagosome is a kind of, now enzyme will digest the food, na? Enzyme will digest the food, hydrolytic enzyme digest the food. After digestion, nutrient get absorbed and go in the cytoplasm. Hello? Digestion after absorption occur or not? Yes, it occur. After this, what's happened? After this, what's happened? This food, the undigested material, is packed in a vacuole, membranous structure, and this is called a residual body. Residual body. Here, who is there? Undigested part is there, material is there, who not digested. Getting the things or not? Everything not digested, na? Getting the things. So it is present in the residual body, and this residual body is also called which lysosome? Tertiary lysosome. Tertiary lysosome. So primary, secondary, tertiary. You got the idea? Primary, secondary, tertiary lysosome. You understand the idea or not? Now suppose that in this cell there is an old mitochondria. In this cell there is an old mitochondria. Another possibility is what? This lysosome fuse with mitochondria. It's not like the lysosome is only one. So this lysosome fuse with which one? Mitochondria. And when it fuse with mitochondria, what happened? Look, when it fuse with mitochondria, it become like this. Here, old mitochondria is at this, and they release the enzyme on this. So who will digest it? These mitochondria will digest it or not? So it is digesting their own cell part. So it is also a secondary lysosome, but it is also called autophagosome. Auto means self. Phagosome. Are you getting the idea? Understanding the idea? Is it clear in the mind? So this is the lysosome, different forms in different physiological state. Understanding the idea? Autolysosome. Getting the things. Understanding the idea? Clear in the mind? Now what happened? What is the function of the lysosome? Function. Intracellular digestion. Wherever intracellular digestion occur, who is there? Getting the things? Lysosome is there. Lysosome function is autophagy. Autophagy means eating or digesting the component of cell. Like eating a component of cell. So it is helping in cleaning of the cell. Heterophagy. Heterophagy means digesting outside of the things. Heterophagy. Suppose that lysosome of WBC eating the bacteria. It is called what? Heterophagy, digesting the bacteria. When WBC do the phagocytosis, they engulf the bacteria, na? And after engulfing, they digest them. Na? Who digests them? Lysosome digests them. So it is digestion of food, you can say. digestion of food inside the cell. Inside the cell. It is called heterophagy. Another function is autolysis. It is digesting the cell. When prolonged starvation occurs, lysosome bursts and cell is going to digest. And that is the reason it is called suicidal bag. Getting the things or not? Suicidal bag of the cell. Because it performs which one? It is associated with which one? Digestion of the cell, killing of the cell itself, autolysis. And that is the reason it is called suicidal bag of the cell. Now, metamorphosis in frog. Also, who is performing a role? Lysosome. How? In tadpole, who is there? Tail is there. And in adult frog, tail absent. So, tail is digested by whom? Lysosome. Tail is digested by what? Lysosome. It is helping the maintaining the shape of your bone. Whenever bone going to grow abnormally, they are eating that cell, osteoclast cell. Getting the things or not? Bone eating cells. You are getting the idea? Understanding the idea? Clear in the mind? So these are some of the function associated with which one? Lysosome. Now we are coming on vacuole. Vacuole. So in how many type of vacuole we will see? Large central vacuole. Then I will see about which one? Food vacuole. Then we will see about which one? Contractile vacuole. So look about these all. Large central vacuole present in plant cell. So in plant cell, you will find more than 90% of the area is occupied by the vacuole. And this is the membrane of the vacuole. It is called tonoplast. And here it is the cytoplasm. And fluid present in the vacuole, large central vacuole, is called cell sap. Cell sap. And this cell sap is hypertonic than cytoplasm. So how it is hypertonic? So there is a pump protein present in the tonoplast, and through this pump protein, many solute is transporting actively by expenditure of ATP. Getting the things or not? Many solute is getting transported actively, and that is the reason solute concentration inside the cell sap increase, and they are hypertonic than cytoplasm. What is present in large central vacuole? So in large central vacuole, water is present. Water is present. Excessive water. Excretory product is present. Excretory product is present, and another things present is that other material not useful for plant. Other material not useful are all present in which one? Vacuole. These large central vacuole. So it acts as a storehouse of the cell. Unnecessary, unwanted, excess thing is present in this vacuole. Getting the idea? So this is the large central vacuole. It is occupying around 90% part of the cell. Around 90%. And this is tonoplast, and fluid is called cell sap or tonoplasm. Now we are coming on food vacuole. Food vacuole, an example, what happened? Amoeba. or a smaller organism. When they go for ingestion of the food, food is surrounded by a membranous structure, and that is food vacuole. So food vacuole, as name suggesting, it containing food. Able to understand? Food which came by ingestion, surrounded by a membranous structure, food vacuole. Contractile vacuole, also where it is present, example, it is amoeba, and these contractile vacuole is associated with osmoregulation. Osmoregulation. It work like human kidney. It work like human kidney. Work like human kidney. In our body, kidney have a role in which one? Osmoregulation. Na? So same it is doing. Contractile vacuole. Able to understand or not? So contractile vacuole have role in which one? Osmoregulation. Is it clear in the mind? Work like human kidney. So have a role in excretion as well. Able to understand? Got the idea? Understand the idea? So this is about what? Vacuole. Done? Is it over? Any doubt is there, you can ask. Kiji, here I not understand. Help me out. So, clear? So, just remain methodical and focused. All three. Yeah. What you are asking? Asking any question? Okay. So clear? Understand? Any doubt? QR code. So just be consistent, work hard and prove yourself. Be a person of a spine. I have some capability and I will show the world. See who am I. Getting the idea or not? That is the most important part in the life. Prove yourself. You not known by the name of your parent after few years, your parent managed to known by your name. And people take introduction, dear parent. That things require in the life. And with the proud, your parents say, my child's. For that, 8-10 months. Life set. Okay, ji. Done. So, should we discontinue the meeting now? Should we discontinue? Okay. Over. Okay, ji. Bye-bye. Have a good day to all of you.